making proteins.
In translation, the mRNA leaves the nucleus and goes into the ribosome, in which the tRNA helps break down the mRNA and creates proteins, which are called polypeptides. In transcription, the DNA creates mRNA inside the nucleus.
DNA replication is the process by which DNA makes a copy of itself during cell division. The separation of the two single strands of DNA creates a Y shape called the replication fork. The two separated strands will act as templates for making the new strands of DNA.
Extraction of DNA. In the extracting strawberry DNA lab, the soap and salt mixture broke open the cell membrane and the ethanol brought the DNA to the surface of the solution.
Restriction enzyme, also known as restriction endonuclease, is a protein made by bacteria. It works by a matching process, and when in contact with the DNA sequence that goes with the enzyme, it will wrap around and cut the DNA in a specific way.
The polymerase chain reaction makes extra copies of the gene. For example, each of the DNA samples collected are individually put through a polymerase chain reaction and are replicated to create more copies for testing. DNA electrophoresis.
DNA is negatively charged and will attract a positive. Electrophoresis is the motion of dispersed particles related to the fluid under the influence of a spatially uniform electric field. Electrophoresis of positively charged particles is sometimes called cataphoresis, while electrophoresis of negatively charged particles is sometimes called anaphoresis.